Welcome. Today we will discuss about the cell cycle, and this is a very important topic in biology. So we all know that we all starts our journey from a single cell, which is known as zygote, and then this goes on multiplying and leads to a multicellular organism. So cell division is very important, and the cell cycle is nothing but the lifespan of a cell. So in a cell cycle, or rather a cell cycle can be divided into two broad phases. One is the interphase, another is the mitotic phase. So in interphase, this actually deals with the growth of the cell, and in mitosis the cell divides. This is the dividing phase. So interphase can be again divided into three phases: the G1 phase, or the first growth phase, and it may be called as gap phase. But I prefer to call it as a growth phase rather than the gap. Then interphase. So this is the next phase will be the S phase, and then again G2 phase, the second growth phase. And mitosis, actually we know that it consists of prophase, prometaphase, then metaphase, anaphase, telophase, and cytokinesis. So there is another phase which is known as the G0 phase. This is the quiescent state or the resting state, or you can say that at a senescent state, where the cell rests. It do not undergo any division. So there are certain cells in our body, like the RBC, then neurons and cardiac cells, which remains in the G0 state. Now, as the cell progresses for division, it first go through the first growth phase, then to the S phase. So prior to progression into S phase, some conditions are to be met. What are these conditions? First of all, the cell cytoplasmic contents should be increased. The cell must contain all the proteins and enzymes which will be required in the S phase during DNA replication. So the cytoplasmic content as well as organelles are formed in the growth 1 phase. Then if all these conditions are satisfied, then the cell progresses from G1 to the S phase. And in the S phase, the DNA replicates. That is, we know that there are 23 pairs of chromosomes in case of human being. So 23 chromosomes we get from our father and 23 chromosomes from our mother, and these 23 pairs gets replicated to 46 pairs. So they will be distributed to two daughter cells equally, since it is a mitotic division. And when the DNA replicates completely, then the cell enters into the G2 phase. This is again very important phase where the cell prepare itself for the division, the actual cell division. Now for division, what are the prerequisites? First of all, there is the need of certain proteins which are responsible for the formation of cytoskeleton and spindle fibers. So when these conditions are fulfilled, then the cell progresses from G2 to the mitotic phase, that is prophase, then prometaphase, metaphase, anaphase, telophase, and cytokinesis, and ultimately give rise to two daughter cells. So whether a cell go for division, the decision for the division will be taken in the G1 phase. Either it go for division, then it progresses into S phase, or it may go for rest. That is, it may remain as quiescent. Now, in this slide, we will just see what happens in G1 phase. Here you can see that these two chromosomes, and they in S phase they gets replicated, and in G2 phase all the spindle fiber forming proteins will be synthesized, and the cytoskeleton, the proteins which are responsible for the formation of cytoskeletons, they will form in this second growth phase, that is G2 phase, and then finally the cell progresses into the prophase and then prometaphase, where the spindle fibers starts reappearing, and then in metaphase they arrange themselves in the equatorial plate. In anaphase they the chromatids start separating in the two different poles, and finally in telophase you'll find the reappearance of the nuclear membrane, and the centrosomes gradually disintegrates, and it will lead to the two daughter cells. Now in this slide we will just see what happens in the nucleus, and here you can see that the nuclear membrane starts disappearing, and the two centrosomes starts moving to two opposite poles, and in prophase you find these chromosomes they starts arranging themselves in the spindle fibers, and in metaphase they actually arrange themselves in the equatorial plate. The spindle fibers gets connected with the centromere, and these two centrosomes they actually move apart to different poles. In the early anaphase these chromatids get separated and starts moving towards the two opposite poles, that is there is pulling effect, and in the late anaphase also you can see that these chromosomes actually moves apart in two different poles, and the spindle fibers get squeezing, and in telophase you can see that there is a reappearing of the nuclear membrane, and finally in deposition and cytokinesis these two daughter cells get separated, and they are released as free cells, as daughter cells. So this is all about the cell cycle. So but there are three vital checkpoints. So the cell progression to different phases, they are checked in G1 and S, and another is between G2 and mitosis, and another itself in the metaphase. So in these three checkpoints, the cells are checked whether the conditions that are required are fulfilled or not. If, for example, if the cell is in G1, it will progresses to S phase only when all the proteins and enzymes and organelles as well as the cytoplasmic content gets increased, and otherwise it will not progress into the S phase. And similarly, from S phase, S to G2 transition is also important. If there is any damage in the DNA, then this damaged DNA should be repaired in the S phase. Then only the cell progresses into G2. And again, another checkpoint is here for G2 to M phase transition. And this checkpoint actually checks whether the cell in the G2 phase has developed or synthesized the proteins necessary for the spindle fiber and cytoskeletal formation. And if it is satisfies the condition, then it progresses into the prophase, metaphase. And metaphase itself, there is another checkpoint which checks whether the APC is released. If APC is released, then the cell will progresses into the anaphase, telophase, and finally it will through cytokinesis give rise to two daughter cells. So this is all about the cell cycle. We will discuss the cell cycle regulation in more detail in another video. Thank you.